Hello, everyone. Matt here at Cybergenetics. Welcome to our, what is this, April TrueAllele user webinar. Today, myself and Bill Allen will be going over a case and an exercise that we have done involving a mass disaster where we do some victim remain identification using kinship. Um, so I was just letting everyone get in, but I think we have a group of people now, so I think we're going to get started. Bill, is there anything else I have to cover before we get started? Are you good to go?
Did you let everybody know that we're recording, that this will be available afterwards if somebody's missing it?
What he said. Okay, so let me just share my screen to get that up, and then we will get started. Okay, let's move that off the side. And okay, so Bill is going to start, and then I will chime in with the demonstration. Pretty much the way this is going to go is Bill's going to kind of present what we're talking about. We have some slides here, and then I'm going to jump into the software relevant points.
Okay, so to get started, this is a talk I gave at APHIS back in 2023. As you can see from the title, this is identifying victim remains using the kinship genotype inverse. How we use TrueAllele to identify victim remains. So if you're going to go to the next slide, Matt. So in 2008, there was a horrific bus crash in South Africa. And I think there was like 80 some people on the bus, and many of them, survived the crash, but there were those who did not. And of these victims, there were 15 victims that were their bodies were burned beyond recognition. And so, how the police wondered when they were recovering these remains, how would they identify them? Because some of these people were from rural communities, so there was not even like dental records. There was nothing. Like again, their bodies were burned, their artifacts were burned. There's nothing. So they thought that well maybe DNA could be used. So they got DNA from the victim remains. And then they also solicited DNA from the community, from like where the bus is, like the bus route. And they said, if you believe somebody, um, that was on that bus and they're still missing, please, can you submit a DNA sample? Also, can you let us know what your biological relationship is to that person? So this also happened, and the police collected that information and they got the DNA. And the lab wasn't able to do anything with this information. They weren't able, I'm not sure like what they intended, but they weren't able to identify any of these remains. So I guess in a long shot, maybe in from their point of view, they sent it to cybergenetics. Cybergenetics had at that time is already like identifying victim remains in the World Trade Center disaster. But they said, okay, why not? Because they weren't able to do anything with that. And if you could go to the next slide, please, Matt. So here we just have a list of the 15 victim remains. This is, again, we received the sequencer files for them. And again, TrueAllele, we just use analyze to get their genotypes. And then the next slide, Matt. We also received 16 people's DNA. They believe that they were family members or somebody who was missing on the bus. Along with their DNA, they also submitted what they believe was their biological relationship to the missing person. So then the next slide, Matt. So the task, I believe, obviously, is to use these people's DNA and their kinship information, how they're related to the missing person, to identify the victim remains. And now Matt's going to show us how that works.
Okay, so you should see my desktop now. And so what I'm going to do at this point now is show on a set of similar data. So this isn't the actual data from the bus crash. This is actually data that is very similar that we constructed for the exercise, which we'll talk about later. And I really have to do four things here. And so I'm just going to put this up as kind of a guide to myself. So I need to first process the victim remains, which will get us the genotypes of the missing people, their physical remains. Then I will provide family references. Once we have these physical remains, we need to figure out who they are. In order to do that, we need a reference to compare to. In other cases, like the World Trade Center, we've used personal effects before. In this case, we're going to use kinship information derived from family references. So I need to get those together, those family references, and then I need to infer from those family references the biological profiles of the probability of the missing people. And then once I have that, I will have genotypes from the remains, genotypes of the missing people, and then we'll need to connect them to each other. And so those are the four steps I'm going to go through here. So to get started, we need to process the victim remains. So what does that look like? So what I have here is just a set of data. So what happened is the lab collected samples from the remains, went in, processed it in a lab, and then sent to us the victim data. And so here, what I just have is a folder of HID files, and it's just literally named victim 1 through 20. And so we're going to try and identify these. And the first step is that we, of course, have to bring it into the system. So the way that we do that is a process that I'm sure most of you are familiar with, which is analyze. So this particular data, I believe, was a global filer from like a 3500. And so we went in and we did analyze on that. Created a data disk, and then the output of that is a set of quality checked quantitative peaks in a .gel file. So I'm just going to, that just takes a few minutes, and I'm just going to, we did analyze, and then let's move to the data module where now we have this victim remains data. So I'm just going to open that data up, talk about what two things I need to do in order to process the victim remains. Okay, so brought in those that raw data, the HID, did analyze on it, converted it to quality check quantitative peaks, which is now in a gel file. And so now, in order to process this, I need to do really just two things. I need to put these samples up on a TrueAllele world, and then I need to ask questions about these samples. So here, these are just the victim remains data. Um, Just going to zoom into one. And so each row here is a different victim remains sample. And the first step is just load it in, send it up to the server. So, that takes almost no time. What I'm doing is I'm taking those quantitative peak data, uploading it to the server so that we can ask question about. Now, typically, in like a casework process, the next step here is then to go in and start asking questions about these data. You go in, you annotate it, you ask, how many contributors do I think is here? And so in casework, this is a process where you might actually look at the data and you're making some sort of conclusions, like how many contributors are there. For more automated processes, though, I don't even need to touch request. Instead, I have these 20 samples. I know they're all victim remains, and then I can process them in one large batch without having to even touch request. So the way that works is that then I go to database upload request and log in. So give me 1 second here. And so what this is doing is this is replacing the request process for all 20 samples with a more automated process where my input to this is just that .gel file. So I've already uploaded this to the server, and now I'm going to use it to form questions. And so what we have here is our batch process window. This is pretty flexible. It was originally designed for like doing database work. So if I were doing like evidence data, where it may be a mixture. I have the option to set it like the three unknown at a short cycle, like 5,000 cycles, and that's how we would do evidence data. In this case, these are just victim remains, and they're mostly single source, so I can either run them as a one unknown or a reference. I can then provide whatever tagging information I want. And the reason why this is important is that on the database, I have different locations that I can send these genotypes to, which are clients. You can also think of them as like bins. And the reason why I specifically want to point these out is because these bins are very helpful in organizing things, for instance, like for databasing. And so on this world, you'll see that I have a general side bin. I have evidence, kinship, remains, suspect, victim. And so I can say, take this entire set of questions I'm about to create and put it in the remains bin. And what that enables for us is as you'll see later, that enables like matching, for instance. I can say, for every genotype in this bin remains, match it to, for instance, all the kinship profiles that I generated. And so that's important. Then I can just say sample information, how long I'm running it for, and how long I'm running it for, and then I just say okay. And then what we will see now is that this has automatically for me gone into that um, gel file and produced the question for every sample, including the positive. I can just check that off. And so in seconds instead of minutes, I can have this entire set of questions ready to go. If I say okay, these would upload to the server just like normal requests, immediately begin solving in a parallel fashion where, if I have 20 things waiting there, all 20 would be picked up and be solved uh, in a short time. And so that is the process of processing victim remains. Um, do analyze, upload the data, and then instead of asking each question individually, ask it as a batch. Saves a lot of time. And so this is very helpful. I know that if your group is using database, maybe you're using this to, instead of having to look at every little piece of evidence that goes up to your database, you're letting the database inform you of where your matches are coming from. And so this is the process you use, a more automated request process. So then once that is done, now we're going to have the genotypes of the victim remains inferred for us. The other thing we need to do is we need to provide the family references. And then from those, we're going to infer the biological people. In this case, this is pretty straightforward. Uh, the biological, the family references were provided to us as text files. So these are just how many we have here? 18 text files, which are just profiles. You're probably familiar with this. If you're not, I'll just open one up real quick. All it is literally a text file showing locus, desig1, desig2, and then the probability is 1 for a reference. So this person is 1010 at CSF and 1314 at D10. And so these are just used where like we have the reference is already known. We don't need to do any processing. We can just provide it as a text file. And for that, again, super simple process. I'm in the data module right now. I can go to database, upload genotypes, point in that folder, and then it will upload the genotype information for this entire set of people. And you'll notice here that it is just being put in the general side client, as indicated by the file naming. So once that is done, we will now have all these family references up there. So the next question is, how do we infer the biological profile of the genotype of these missing people? And so for that, I'm going to refer back to Bill for a minute.
Okay, Matt. Just to reiterate, like what you did. So basically, you just analyzed when you had sequencer data and used batch mode to upload the victim remains profiles, and then you uploaded the text file genotypes. So you have the victim remains row, and you have the family member sample row up. So, all right. So just talk a little bit about kinship. You're going to go to the next slide, please, Matt. So in this process, what we're trying to do is have the computer infer the genotype of the missing person, up to probability. With more kinship information, you'll get a more informative missing person genotype. So here we have, you see there's that dark square there in the middle. That's our missing person and how they may relate to other family members. If you look to the left, we have siblings. Above that, we have parents and grandparents. And below that, we have children. A spouse is not biologically related, but when you have children, then they are part of the equation. So, like in my particular case, I have four children. If you infer my DNA profile from one of my children, well, it's going to be somewhat informative. Add a second child, add a third child, add a fourth child, add my spouse. So then you can kind of like take her contribution out of this, and you'll basically be getting um, my genotype from that information. Similarly with siblings, if you use my siblings, use my parents and my siblings, you're using all this information. So you can add all this information together to get like a very informative genotype. But in this case, with these victim remains, we only had one. And sometimes it wasn't just somebody who was just like one generation, like a parent or a child, like or a sibling, like very closely related. Actually did have like grandparents and grandchildren. So the computers have to fill in, and still use what information it has to find or to identify the missing person's genotype. So if we just want to think about that, if we can go to the next slide, please, Matt. So you can see why this may be an issue because I assume I'm speaking to people who've heard of a Punnett square. Going from parents or knowns, and you can infer like a person's or like a what your child or what a child could be up to probability, based on the parents' genotypes. But if you go to the next slides, please, Matt. How do you go backwards? How do you go from, say, a child to his parents, or a child to his grandparents, or grandparents to a grandchild, or siblings might be, but how do you work that out? And again, how do you do it not only at like one DNA location, but all of the DNA locations? And that is the power of the computer here. It's just going to do this, and it's just going to do all of this automatically. And I think the next step is Matt's going to show us how it does that.
All right, so what we're gonna do here is now we are going to infer those missing people. And so for that, as Bill mentioned, we have those family references up. We know their biological relationship. So effectively, on the pedigree chart, we know what the connections are, but how do we provide that to the system? Um, and so the way that we do that is using a kinship table. So I'm just going to open this in Excel so it's a little bit more readable. And so what this text file here is it's really just a text representation of the information from the pedigree. And so what we have here is each row is actually a separate independent genotype that's going to be inferred, and that's based off of the family name here. And so effectively, the family column is how are things grouped together. In this case, we only have one relationship per sample. But we could have, their kids, their spouse, their parents, in which case there would be multiple rows for the same family. And so that's the first column. Second column relationship is that connection on the pedigree chart. Is it a child? Is it a sibling? And this is all relative to the missing person. And then next, we have the client, which is the client where we have,
Matt,
oh, yes?
I'm sorry, quick question. The relationship is based on to the missing person or what is the provided?
It is based off of, so let me go back to the slide real quick because that'll make it easier to answer this. It is based off of the missing person. So the names here on this slide are what is entered in that table. So
So
for
the missing
instance,
person is the child.
the missing person is actually the parent.
Okay.
So
So,
like these entries here on this table are the entries here. So like child one, I'm saying that this person, child one, is family member 21. So family member 21 is this.
okay.
And the where you can really see that is like here for a grandparent. Mother M is the um, maternal
Yes.
So
grandmother.
that's what was provided. That's
Yeah,
actually goes by the relationship of the reference provided by the relatives.
yes.
Okay,
So
got
it's
it.
front, so everything focuses down onto the missing person. So like if I had the grandmother on the maternal side, it would be Mother M. Then if I had their mother, then if I had their child, so then the three entries would be Mother M, Mother, and then Child One, because
Okay.
everything's relative to that gray in the center.
Thank you, Matt.
Yeah, no problem. Which is actually probably the most complex part of defining the kinship relationship is remembering how this is all related, which is why in our kinship manual, we actually have an extended pedigree like this, so you can just look it up.
So,
So
by
if
the
you,
way, do you guys use any software to build the pedigrees or
nope, we just, once we know what it is, what the relationship is, this is the representation of that.
of that? Okay, got it. Thank you.
Yep, no problem. Yeah, so actually, I'll leave this up just because it'll make this a little bit easier. And then let me slide this over. And so here you can kind of see what we did. So for this family, we had the missing person's child that gets entered in here. And then these two last columns are just the coordinates on the system where that family member's genotype is. So whenever I set up that request, if I go back and we look at those text files, you'll notice that it's family member 21 is the name. And so, and we uploaded it to SIBE. And so that's what this is pointing at. And so then what will happen is that the system will then use this information to go do the inference process for us. I'll talk about that in just a sec. Just work through a couple more things here. So, different family, we have their sibling. And so that is the sibling of the missing person. Here, Father M is the maternal grandfather. And so we have this person, and it is the grandfather of the missing person. Sibling, father. And so these entries on the relationship are just defined by this pedigree. And so literally when you're doing kinship, you just look at our kinship manual and you can just be like, okay, I have father, their father and their mother, so this is what I enter in. And so then once that is done, what happens is I then take this kinship file, go into the data module, and then I say upload kinship. And then what will come up is a upload window here, where really I'm just doing two things. One is I'm saying whenever I make these kinship genotypes, so these will create new genotypes based off the family references. Where do they go? And so here I might put them in the kin client, but I have the ability to put them wherever I want. So for instance, if I was using this database and I, wanted to do some familial searching and I said, okay, I have this person, but I want to infer the genotype of the brother, maybe instead I'm putting that kinship profile in suspect. So we're not limited where these genotypes go or how they're used. If you want to, if you have a set of suspects and you want to search all their siblings, you can have the system automatically do that for you, and then put those genotypes in as suspects, and then search those as well. And so, just talking about like other aspects of how this can be used. And then the last thing is I just need to point at that file. So I have my kinship file there. And so with those two things, what will happen is I click upload. This goes out to the system, and then what it will do is based on these relationship, it'll do the probability math to figure out what the genotype is for each of these people. So for example, in our question here, where we know the child is 11, um, 12, for one of the parents, I may say that, okay, there's, you know, a 50% probability of them being an 11, and then you spread that over the possible alleles, and then a 50% from the 12 and spread that over the possible alleles. The idea, though, is that all this working backwards, is just genetic probabilities. So the end result of this is a genotype that is a list of allele pairs with probability. And so that is exactly what we get when we're referring from evidence data. And so the system can treat it the same way. The result of inferring kinship is I'm just going to get a list of possible genotypes with probabilities associated to them, and those look just like our evidence genotypes, and they can be compared and matched just like anything else in the system. And they can also be, you can you can do fun complex stuff, infer someone's sibling and then match it to a minor component of a five-person mixture that you inferred as evidence. And it's a probability genotype matching to a probability genotype. The system doesn't care. Um, and so then the result of this is that 20, or what do we have here? 18 genotypes are inferred based off of the family uh, references, and now we have a probabilistic profile for all these missing people. And so we have some set of genotype probabilities that is the biological relative of these people. And so now the last step is to identify these victim remains. What is the connection on that two-column slide back here? How do these link to each other? And so the way that is done is that in the system, and this is why the bins, the clients are very helpful. In the system, I can set up automated match rules. And so what I have here is that on this database, I have Evie to Sib, Evie to Evie, Evie to Sus, and remains to kinship. And so what these rules do is just by them existing in the background, the TrueAllele system is saying every genotype that I see in evidence automatically compare to every genotype in SIBE, and then based on some limit I set, if I see a positive match statistic above a certain level, store that, and then just leave it on the database, and then I can come in and harvest those matches. So same for evidence to evidence, and more importantly for this, same for all those remains genotypes matching to kinship. And so literally, it's just a rule I set. I say I select one bin and I select another bin and I say make that match, and then everything else happens automatically. I'm not like going in and like hitting a button to make it happen. It just all I'm doing is producing genotypes in those two bins. And so then what I can do and what that enables for me is that I can come in here into our report module where typically, if I'm doing casework, I'm going to come in, load up my evidence genotypes, load up my subject reference, load up whatever populations, and then look at those comparisons, maybe look at the data, that kind of thing. However, the system has already for me compared everything in remains to everything in kinship and knows what the actual associations are. So instead of finding genotypes, I'm going to find matches. And so whenever I do that, it brings up a query window that lets me search very specifically. So in this case, it's going to be pretty broad. I'm just going to say, okay, for everything on the database remains the kinship, show me any match higher than 10,000. And so that's what I'm about to do. However, I could get a lot more specific. I could set time limits on this and say only show me stuff like within the last week. I could say I only care about one specific piece of evidence, and I could fill in that information as either genotype 1 or 2 and say limit the search only to things that match this. But in this case, we just want to know for any of our remains, did they match to any of our inferred kinship genotypes? And so then I say, okay. And so then what I get here is I am getting a list of matches. And so normally we have a genotype and we're matching to another genotype. Here, that match has already happened. And so what I have here is a pair of genotypes. On the left-hand side, I have victim 14, who is in the remains client. And then on the right-hand side, I have the kinship genotype for 30, family 34, and they're matching to each other. And so that's the score in the center here. And so this score is a database match score, which is using the psi population at 0% theta. However, as we'll see in just a second here, I can load in this information and then compare it to any population I want. But what this is giving me now is that I have here 12 matches where I have a left genotype, victim 15, matching to family 35, victim 3, matching to family 23, and then these are the match scores, ranging from a billion up to um up, up to
10 billion billion.
yes, thank you. 10 billion billion. Uh so up to the 10 to the 19th here. So here's a trillion, there's a quadrillion, and so on. And so I have all these matches and they're relatively strong here. And so then I can load them into the system. And so what this will do for us is this will load them into the system in genotype pair mode, where typically when we load things into report, we're looking at every piece of evidence across every piece of reference. Well, here we're looking at specific matches. We're looking at a set of paired genotypes. And so whenever we see it, what's going to happen is the Q1 evidence is going to line up with the S1 reference. And you'll see that like everything is paired with each other. So this will just take a second to download from our server because I'm just actually connected to the cloud server and it's downloading right now. And then once that comes in, we now have our victim. These are our remains. These are our inferred kinship. So here's our two columns again. And then I can just add a population to compare them to. So I'm just going to use NISTAL, large multi-ethnic population here. And so now theta would be applied because I have a default to 1. And so I can see in our match table, instead of our typical cross table, now what we're looking at is a 1-to-1 match table. Victim 14 matching to family 34, the kinship genotype inferred from that, at a stat of 10 to the 17th. Then I can hit this. And so I can see quadrillions, trillions, whatever the case may be. And so that's all accounted for using our typical match calculations. And so what this gives to us now is these are the connections between those two columns. And it was all generated automatically. I'd never, you know, I other than analyze, I didn't like skip any steps in the process. It's a matter of, doing these more automated steps where I'm just asking 20 questions at a time or uploading a set of profiles, uploading a set of kinship relations, and then the output is matches. And so what does that look like? And for that, I'm going to hand it back to Bill.
I'm just going to keep going down a few more slides. Matt, I'm going to go to the 12, please. Okay. So here we are. Here's what we were trying to, the problem we were trying to solve. We've now connected, as you can see, with very colorful lines. We've connected victim remains to samples or family members. So this is the actual results from the bus crash from 2008. And you can see, I think we got 9 of the 15 victim remains identified. And something we learned because we're like, well, why didn't we get all 15? Or what happened? Was there something going on? One thing we learned is culturally, some of you may have reported a bio or a relationship that wasn't actually biological in nature. So somebody may have adopted a child, and that child was their child, or that was their parent, though there was actually no biological relationship. So even though they would say, "This is my son," or "This is my mother or my father," so there could be that. Again, maybe some of these people were not their family member didn't contribute DNA. The one interesting thing, or I'd say more interesting thing, was if you go to the next slide, Matt. We actually had somebody submitted their DNA to the police, but did not give their biological relationship. They just said, "Well, I believe my family member's on the bus." So there, all we did was we just had Trullo infer the parent child and the sibling. And on that sample, we actually got a match as well. So it didn't really even need this other information, though it was helpful. We could have just done that automatically for everything, just as, infer every parent, child, and sibling automatically, and then just compare. So if you go to the next slide, please, Matt. So you can do this. And you can say, well, you're just saying that. But first of all, we are going to offer that as an exercise. I believe this is part of the training that we do now. I'm training analysts, but as part of this webinar, we'd like to make this available for anybody's interested, like to go through these things. And you may be like, well, that'd be fun, but I don't have the time. You do have the time, and I know that. And the reason I know that is we can go to the next slide, please, Matt. So Dr. Pearl and I were in India, and he had the idea. He's like, Bill, can we just do this with the students we were training there? They were just giving a course on DNA interpretation. These students had seen, this is like the third day of seeing TrueAllele, and I mean, like, they saw a report like the first day for a half hour. They saw, like, these are not people who are trained in TrueAllele. We said, can they solve this problem with just some basic training? So if you can go to the next slide, please, Matt. So what we asked them to do is do analyze, take the sequencer files, get that gel file, use data and batch mode to infer the victim genotypes. Do the same thing with the family remains. Use data to have TrueAllele infer the kinship genotypes. And there were 20 students, and took a little bit of math because we decided to do this the night before, and we had a limited server of just 12 processors. Could all of the processing be done over lunchtime? Because all it has to do, the reference genotypes are very quick. When TrueAllele infers a kinship genotype, that's like instantaneous. The matching is basically instantaneous. So could all this be done? So that's what we did. So we had all the students run analyze, do all those steps, upload the genotypes, upload the kinship, have it match while we sat and ate lunch. And then when they came back, we said, did you solve it? And they were able to. They were able to use, like Matt showed, find, match, and identify the victim remains. So I'm saying if you have a little bit of time and you'd like to go through this exercise, you could do that and it would not be a burden. So we go to the next slide, please, Matt. So again, we just wanted to show through this is, again, with the TrueAllele you already have. Again, in these mass disasters, you're able to do this. It's automated, it's informative, and again, I'm saying this from the experience that it is easy to use. And anything else to add, Matt?
Yeah, no. Just that yeah, we are planning on doing a group session. Probably, I would guess that just based on how long it takes to schedule, a bunch of people, probably next month. But if you're interested in just running through this exercise, it would probably be like it'll be a few hours probably of we'll solidify it, but if you're interested, just reach out to me, Matt L at cybgen.com or Matt at cybgen, or if you have any like your trainees that you would want to do this. And we're just planning on doing this as like kind of an add-on because we're still planning on a different webinar for next month as well. And so if you're interested, just reach out to me. Let me know. I'll get you on the schedule, and then we'll loop in as a group. So, um, and so then, just to kind of recap, the main idea here is that we had multiple things working in concert here from TrueAllele, things that are built into your current TrueAllele system, database and kinship. And so um, we had victim remains for those genotypes. We had family references, used the kinship module, which is built in to infer the missing people, and then using the built-in TrueAllele database, connected those two, the remains to those inferred kinship. And so this is using pretty much a lot of the like built-in stuff of TrueAllele that you might not necessarily know about, using both kinship and database together to solve this problem in a straightforward fashion. In some ways, it's almost easier than casework because the computer is doing a lot more work than for me in this process. Um, and so with that, I think we can open the floor to questions. And if you have any questions, feel free to either put it in chat or just ask. And otherwise, thanks for attending today.